Okay, we've only got 3 people here right now, but I'm assuming that everybody else who's in this course will eventually hear my sweet dulcet tones. Because only 3 people are here, I'm seriously thinking about ordering some pizza and maybe a cake, but I guess you'll miss it. But I got to talk a little bit first. So I want to finish talking about Sympatric speciation. And we've already talked about how it can happen when you have the possibility of chromosomes doing weird stuff, like polyploidy or hybridization, both of which can happen in animals, even in a few vertebrates, but is surprisingly common in the evolution of plants. The question used to get asked a lot, was it possible for speciation to occur not by chromosomal hijinks, but just gradually by good old-fashioned variation plus selection without a geographic barrier, without a population getting split by either by dispersal or vicarious? And up until probably about 30 years ago, see, I'm 52, so my definition of a few is not necessarily your definition of it. The stock answer used to be no, but we now have some pretty well documented cases of gradual sympatric speciation in progress. And this is one of them. That's a fruit fly called Ragoletus pulmonella. And it's affectionately known as the apple maggot fly. And it's a pretty severe pest in areas that grow apples. The adults lay their eggs on apples, and the larvae burrow into the apples and eat them. They can, by the way, and this will be important in a minute, they can attack other fruits as well. I think they can go after berry crops and things like that. But the larvae will feed on fruit, and I'll dim the lights just so you can see that picture. That's what one apple maggot can do right there. When people are talking about finding a worm in an apple, that's often what they really mean. You know the old joke, what's worse than finding a worm in an apple? Finding half a worm in your apple, right? So once the fruit fall to the ground, the mature larvae crawl out of the fruit and burrow into the soil and form pupils. Like the cocoon or the chrysalis of a butterfly, they form a pupal stage, and the pupae will just spend the winter in the soil. The adults will emerge in spring or summer and mate and lay eggs on the next crop of apples, and round goes the great circle of life, and it moves us all. Incidentally, not that any of you hearing my voice, as far as I know, are particularly likely to become fruit growers, but fruit is actually probably the most pesticide-intensive crop that we grow, just because buyers these days have this expectation that apples, for example, all have to look absolutely perfect and perfectly glossy, that they all have to look like wax fruit. And to enable that, you have to spray an awful lot if you're a commercial apple grower. If you ever become a fruit grower and you want to reduce the amount of pesticide being sprayed, which you probably should, a very simple countermeasure is simply to be proactive about picking up fallen fruit. So if you can pick up your fallen apples before the maggots can crawl out of them and pupate in the soil, you can break the life cycle of apple maggot fly without using chemicals. Never let it be said that I do not teach useful information. You know, that's good. That's actually information that can give us all a more healthy food supply. Right. The adults lay eggs on a bunch of fruits, but in Michigan, where this has been studied, ever since at least the late 80s. There are now two main races of Rhagoletis pulmonella that have been established. They're the apple race and the hawthorn race. And they're named for their favorite foods. Apple Rhagoletis feed on apple, hawthorn Rhagoletis feed on the hawthorn, which is also in the same family as apples. I don't know if you've ever eaten. What was that? Okay. Don't know if you've ever eaten hawthorn, but the fruits, I don't think they taste good fresh, but you can make them into jelly. It's rather nice. Mayhaw jelly is a local specialty in places like northern Louisiana. Anyway, so you've got Rhagoletis that feed on apple and Rhagoletis that feed on hawthorn. And over most of the north central US, the two are sympatric, meaning they share a range. They live in the same range. And it turns out the difference between the two is a difference in timing. What you're looking at is just the lines give you the time in which the flies emerge. So apple Rhagoletis adults will begin to come out of the soil in mid-June, and they will hit their peak in about mid-July. That happens to be exactly 6 weeks before the maturity of the host fruit. Apples in northern Michigan will begin to ripen about the beginning of August, hit their peak in late August, and be done by late September. At least they did back in the 80s. I'm sure global warming is playing merry hell with this, which would be an interesting thing to study. But anyway, the apple Rhagoletis adults emerge about 6 weeks before their host fruit does. Hawthorn, it's the same. Hawthorn flies begin emerging in early August, peak in mid-September, and are done by mid-October. And 6 weeks later, between early September and mid-November, is when hawthorns mature. So there's a separation between these populations, the apple Rhagoletis and the hawthorn Rhagoletis, but it's not a separation in space. You can find them in the same orchard. It's not a spatial separation, it's a separation in time. The apple Rhagoletis adults emerge early in sync with when their food source, the fruit, the apples are going to be ready. And the hawthorns emerge late in sync with when the hawthorn fruits are going to be ready. And you'll note there's very little overlap. There's only a little time in mid-August when both apple and hawthorn flies would be emerging. And few of those are emerging at the same time. Most apple flies are never going to see a hawthorn fly. So we've got two populations that are splitting because they've got different timing and they're specialized for different food sources. We know that hybridization is low, it's roughly 6%. Hybrids suffer reduced fitness. One problem is that hybrids actually emerge at a time that's neither in sync with the apples nor with the hawthorns, so they can't get a lot of food. We know that there is very little interbreeding between them because allele frequencies are quite different at all loci studied. We actually know that the apple flies are not perfectly adapted to apple. Because if you take some apple fly larvae and feed them hawthorn, more of them will survive. Hawthorn is better nutritionally. However, apples are a lot bigger. So it's less high quality food, but there's more of it. And that means more protection from competition. And because the apple flies can burrow more deeply, they're less likely to get hit by parasites. Specifically those parasitoid wasps that lay their eggs in insect larvae that we've already talked about. The apple maggot flies can burrow more deeply and stay out of range of the parasitoid egg-laying wasps. So apple has some advantages, although purely nutritionally, it's not the best food. The reason that matters comes when you look at the timing of all of this. Apples aren't native to North America. Hawthorn is. So before roughly 200 years ago, the ancestors of these maggot flies were presumably living entirely on hawthorn. But apples were introduced roughly 200 years ago as European settlers began to move into the old Midwest, which is now the East, but began to move into Ohio and Wisconsin, Illinois, Indiana, and Michigan, bringing apple trees with them. Early settlers grew a whole lot of apple trees, incidentally, mostly not for food. Most of the apples were small and bitter and didn't make very good eating. What they were growing them for was to make alcoholic apple cider. And this incidentally explains, anybody hear the legend of Johnny Appleseed? Disney made a film about him. He's this cheerful, happy-go-lucky guy who goes wandering around giving out apple seeds to everybody and singing, "The Lord is good to me, and so I thank the Lord." Anybody? This is a Disney film? You can't tell me you haven't seen it. Okay. All right. Yeah, Disney made a version of this. The real Johnny Appleseed was this religious, I won't say religious nut, but he was this kind of, zealot for this oddball group called the Swedenborgian Church, who believed that its founder had visited heaven and basically taken a guided tour of the whole place. And so he'd wander around spreading the Swedenborgian gospel as well as handing out apple saplings and seeds, so that the early American settlers could have their alcoholic apple cider, because that's important. Anyway, Johnny Appleseed and people like him were the ones that brought apples to northern Michigan, and they only did it roughly 200 years ago. Which means that any change that we see between the apple maggot flies and the hawthorn maggot flies, any difference has only appeared in the last 200 years, because before 200 years ago, there weren't any apples up there. So what we seem to be seeing is a case of incipient speciation, meaning it's speciation caught in the act. That these apple and hawthorn maggot flies, they can still interbreed. They may not be entirely separate species yet. This is another one of those cases, right? Remember, it's going to be on the font. There we go. There's an answer to a question that's going to be on the font. The answer is UNH. So maybe we don't have fully separate species yet, but we're watching it in the act of happening, and we've got a timeline for how long it's taken. And so far, it's only taken 200 years to get these populations that are separated from each other, not in space, but in their niche and in timing. So yes.
So did hawthorn and apples, like you said, they're in the same genus, right?
They're in the same family. Hawthorn is Crataegus and apples are Malus, so they are in separate genera, but they are in the same family. They're in the rose family, I believe.
So they would have like diverged, if we're thinking about like statistics way back in like the genome, right?
Oh no, not that long ago. We'll actually get to plate tectonics, although I'm not quite there yet. But no, they would have diverged much more recently than that. There's a number of genera that have members in North America and Europe and Asia. Dogwood is the first one that comes to mind, genus Cornus. And I mean, on and off, I mean, there was a land bridge to Asia up until roughly 11,000 years ago. And I can't remember the exact moment that North America lost its connection to Europe, but it was maybe 30 odd million years ago. It wasn't immensely long. So yeah, I'm not sure what the divergence date would be between Malus and Cretaceous, but yeah, it wasn't nearly as far back as Pangaea, which started breaking up roughly 200 million years. Okay, yeah, good question, but I'll get to the timing of all of that later. It's been proposed that sympatric speciation explains the origin of some other wild species, like, for example, the blueberry maggot fly maybe came into existence in the same way. You got a population that colonized blueberries and became isolated, not in space, but in timing and in niche. Some other studies that began in the 90s have looked at sticklebacks. And this is another case where we've got a pretty good constraint on the time frame for this. I talked about sticklebacks a while back as a great example of sexual selection on traits that are markers of good genes. You might remember that the redder the male's belly, the fewer parasites its offspring develop. Yes, right? Okay, well, these are the same fish, and they've been studied in lakes in British Columbia. And we happen to know that about 12,000 years ago, British Columbia was covered with an ice sheet. That was the highest extent of the last major ice age. We didn't get glaciers in Arkansas, but we did get ice coming down all the way to Des Moines, Iowa. So just on the far side of Missouri is how close the ice got. We've still got some Arctic type plants in Arkansas that were more widespread when the climate used to be quite a bit cooler. And now they're restricted to places like right in front of the mouths of caves, where they've got a steady supply of natural cold air year round. So we've still got a few plants that you normally find much higher north, still hanging on in cool pockets in the Ozarks. Anyway, so 12,000 years ago, those lakes didn't exist. Once the glaciers retreated, the lakes formed, and sticklebacks are tough enough they can live in marine water or in fresh water. So we think sticklebacks migrated upstream from the ocean to colonize these lakes. These lakes would all have, rivers leading to the ocean, and sticklebacks would have just migrated upriver. So on the one hand, you would expect to see separate forms in different lakes, and you do. But what's more important here is that the lakes have, a typical lake will have two different varieties of stickleback in it. It will have a limnetic stickleback, which lives up in the water column and feeds by, for example, picking insects up off the water surface. And there will also be benthic sticklebacks that live down on the bottom. And in fact, in a given lake, limnetic and benthic sticklebacks actually look different. There's a limnetic up top and a benthic on bottom. I mean, they're still visibly sticklebacks, right? But you can tell the stickleback that's limnetic is seemingly thinner. The mouth opens at a steeper angle. I'm assuming that's got to do with the lifestyle that involves picking insects off the water surface, grabbing, I've just forgotten what they call them, those insects that skate on the surface of the water. Hemipterans in the family Garridae. Water striders, that's it. Yeah, water striders. You can tell the limnetic stickleback is spikier, the benthic stickleback has fewer spines, different shapes of the jawbone. I mean, still sticklebacks, but detectably different forms living in the same lake. There's no barrier that physically would stop the limnetic sticklebacks from swimming down and hanging out with the benthics, or the benthics from coming up and hanging out with the limnetics. They can interbreed. You can make them do it in the lab, and the hybrids are perfectly healthy and they're perfectly fertile. So maybe we don't have two separate species yet, at least not by the biological species concept. However, behaviorally, they're isolated because the courtship behaviors are very different. And males of one strain will actually attack females of the other instead of mating with them. So while you can make them interbreed, in the wild, they rarely do. In one particular lake that's been studied, Paxton Lake, only about 1 to 2% of the fish appear to be hybrids. So I guess when 1 to 2% of the time, mom and dad manage to stop fighting long enough. But it's rare. They're not interbreeding very much. They're behaviorally separated. They look different, they feed on different diets, and they select different nesting sites. And I don't have details on this, but apparently there may be speciation by reinforcement going on because wild hybrids appear to have be selected against. Even though they are fertile, hybrids have less chance of passing on their alleles. So we may have selection that's tending to push the limnetic and the benthic strains farther apart from each other. So this looks like, again, this is another case of sympatric speciation in progress. Again, we might not have two obviously separate species yet. This is another one of those cases, but we do have two populations that are distinct from each other and partially reproductively isolated and rarely interbreeding, despite the fact that there is no physical boundary between them. There's nothing physically blocking a limnetic to go down and hang with the benthics or vice versa. And yet we're getting these populations different and probably getting increasingly different from each other. So this seems to be another case where sympatric speciation has been caught in the act. A more recent example, there's speciation in Baltic Sea populations of European flounder. Quite tasty, I'm told, but looks really weird. If you know flounder, they start out as normal fish, but as they develop, they begin to start lying on their sides. And as they develop, the skull develops a twist. And in this species, the left eye actually migrates around onto the right side of the face. And the mouth develops a twist. The whole skull does this sort of thing. Those of you just hearing my words can't see what I'm doing, but it's very funny. And adult flounders spend their lives lying down in this species on their left side, although there are other species that permanently lie on the right side. Camouflage very well. Many flounders can change color, and just hanging out, I think, is just sit and wait predators, waiting for something to come along that they can grab. So, thus the flounder. This used to be the Baltic flounder, Platychthes flessus. And here's the thing, the Baltic is mostly cut off from the ocean. It is still part of the ocean, it's not a lake, but there's only a very narrow connection between the Baltic and the North Atlantic. If you look over at that number 20, right there at the far lower left, that is the That's the Skagerrak Straits. Or shoot, there's another name for it. What's the kingdom that Ragnar Lothbrok rules in the series Vikings? Oh, it's Kattegat, right?
I believe so.
Okay. 20 is about where the real world Kattegat is. The kingdom of Kattegat is fiction, as is most of Ragnar's life, probably. But the Kattegat is that narrow passage between the northern tip of Denmark and southern Sweden, and that's the only connection with the Atlantic. So the Baltic Sea does not, I mean, there is a connection, but it doesn't freely blend with Atlantic waters. And there's also a whole bunch of rivers flowing in, especially in spring when all the snow melts and turns to water and rushes downstream and ends up in the Baltic. And so the salinity of the Baltic, at the Kattegat, it's 20 parts per thousand, which is still pretty salty. The world average is 35 parts per thousand. That's typical salinity. But far away from its connection with the North Atlantic, up in the Gulf of Bothnia and up by St. Petersburg, right about there, the salinity is less than 3 parts per thousand. That's almost fresh. So the Baltic overall is not as salty as the main body of the ocean. And in areas that are far away from that connection, where there's lots of fresh water flooding in, especially every spring, it is almost fresh water. Yes.
What's up with the like the coloration of the shores? They're like green and orange and yellow.
Oh, let me get to that in just a minute. I think that's got to do with ranges of different varieties of flounder, but yeah, I think that will be explained. Incidentally, in case anybody does not know, Lithuania is right there. This is Kaliningrad, the tiny little piece of Russia that nobody actually wants. But Russia's got a big Navy base there. Lithuania is cut off shipments of certain goods to Kaliningrad, and Russia has been threatening to do this and cut off Lithuania through this little pocket right here called the Suwalki Gap. I don't know if you've been following current geopolitics, but this is an area that people have been looking at because Russia is, well, frankly, it's just being Russia. Anyway, I digress. Flounder eggs won't float in fresh water, but they will in salt water. It's a density thing. Flounder eggs are just too dense to float. This, by the way, are stages in the embryonic development of a flounder. Incidentally, you can see if you look at the latest stages down below and towards the right, they look perfectly normal. They hatch as normal-looking larvae with 2 eyes, one on either side of a symmetrical head. And it's not until later that one of the eyes literally migrates around to the other side, and you get a fish that kind of looks like Picasso painted it. Anyhow, so flounder eggs float in salt water, and flounder in salt water produce eggs that float in the open ocean. And this, by the way, is called pelagic spawning. In the fresher parts of the Baltic, flounder lay eggs that stay on the bottom. That's called demersal spawning. And demersal, by the way, means something that can possibly swim, but spends an awful lot of time sitting on the bottom. So a flounder would be a demersal fish. Because it can swim, but it usually sits on the bottom or cruises just over the bottom, and it can spend long periods of time stationary. It's not up there in the water column, you know, swimming around with all of the, I don't know, mackerel or whatever they have. Okay, so that salinity difference means that there are differences in where the eggs are found. the difference between, okay, the difference in salinity creates differences in where the eggs go. And that creates a barrier to interbreeding. Demersal flounders are almost never interbreed in the wild. The graphs that you're looking at here are the results of a type of analysis that we're not really going to have time to get into, called principal components analysis. And in PCA, principal component analysis, let's just say that this is a way that you can take a whole bunch of numbers and boil them down into a limited number that express most of the variance in your sample. If I measured, let's say I made 20 different measurements on a whole bunch of people, I can't plot them all because we don't have 20-dimensional graph data. I think we usually do, but the bookstore was out this morning. But yeah, you can't realistically plot every last one of them. What you can do is feed them into software does this very easily. So you feed them into the R package, and you boil down all those 20 numbers for every person into a smaller number of principal components that express the largest possible fraction of what makes all these people different from each other. So you're boiling down a whole bunch of numbers into a manageable set. And they've done this here with allele frequencies. And you can see that those red dots, if you look at diagram A, where it says second principal component score is graphed against first principal component score, what those numbers mean are that if you take a bunch of allele frequencies of different flounder population types, the frequencies over a very large number of those alleles are very different between demersal flounders, which are shown as the red dots, and pelagic flounders, which are shown as the blue dots. And I confess I forgot what the NS and the TZ flounders are, but you can tell they're quite different from most of the demersal flounders. So the point is that they almost never interbreed in the wild, and allele frequencies at a great many loci are very different, and that suggests minimal gene flow. There can't be very much interbreeding going on, or else we'd see the red and the blue dots intermingled an awful lot more than we do. There's some evidence for selection on demersal flounder genes that are connected with adaptation to salinity. Remember, the demersal flounders are the ones that are living in the more brackish, less salty areas, whose eggs are sitting there on the bottom. And there seems to be selection for some genes that adapt them to lower salinities, as compared with their kin that would live in the ocean and have less and prefer more saline water. And incidentally, the demersal flounders have recently been given a new name because even though they don't look different, they do seem to be a new species. You know, we've got a population that rarely interbreeds with the other population. These were named Platyctes solendali. And we've got some constraints on timing on this because the Baltic Sea, well, first of all, it was completely frozen until about 11,000 years ago. As the ice retreated, it for a while was a giant freshwater lake. It did not break open to the world ocean until about 8,500 years ago. Before that happened, there could not be any flounders in there at all, right? Where would they have come from? I mean, you go from a glacier to a meltwater lake with no outlet. The flounders simply can't get in until there's an opening to the ocean. So the speciation of flounders must have taken less than 8,500 years. And this is one of the fastest cases of speciation ever seen in a vertebrate. And hopefully this is giving you an idea of the timescales that we're talking about here. 8,500 years seems like an incredibly long time, but there were fully modern people living around the Baltic when this happened. I mean, they wore skins and mostly hunted for and fished for a living, but they had sophisticated language. They didn't look grossly different from you. You know, people have been around the Baltic through the entirety of this process. Compared to the total length of time the Earth has been around, this is practically an eye blink. And it suggests that speciation can happen faster than we used to think it could. And that is actually a decent spot for a break. I've got 9:53. So I am going to stop recording and get caffeine.